Okay, welcome every one of you to this our wonderful stay participant. Okay, so I use this one to at least add people. Um, Welcome to every one of you for Biology 205 second lecture. I know we had the first one, which was just a mere introduction to molecular biology. So today, we are going to go a little bit deeper into our lectures, to try to understand a bit about molecular biology. If you look at our course outline, the catalog indicates that today the first part of it was to review the cell and get to know a little bit about the cell, and everything that make up the cell. Because like I said, the whole thing about this molecular biology is just to understand um, that is the way in which genes are synthesized, proteins are synthesized, and what are the functions of these genes and these proteins within our bodies. So that's what we're going to try to look at today. But first of all, we'll try to go a little bit into the historicity of DNA. How was it identified as the genetic material? Because that's very important. So let's get to it. So DNA, first of all, was extracted from. There was a series of experiments that were firstly carried on to determine if DNA is actually the genetic material. One of the experiments was the Frederick Griffith experiment. And, if you refer back to our previous lectures, you know that we discussed some of these things. So it's just going to be a short review for us. Hopefully you can grasp it. So there was a huge debate as to whether DNA was actually the genetic material or protein was, and that was largely in part of the size of DNA. DNA was a lot smaller compared to that of proteins, and proteins have so many elements compared to nucleic acids or nucleotides. That's why the debate was into whether DNA was actually genetic material or proteins. So now, so the first experiment that was carried out to investigate as to whether DNA was a genetic material or not was the Frederick Griffith experiment. And what Mr. Griffith did was that he was isolating this bacteria, Streptococcus pneumonia, from an infected person. Like, I took that particular specimen from pus, right? It removed that from an infected person, an injured person, tried to isolate it, and what he found was something called a nuclein in that particular pus. So he realized there was some kind of infection that was going on, and then he was wondering what was the reason for that particular infection. So Mr. Griffith experiment with Streptococcus pneumonia demonstrated that a transforming substance was used as the replicator. Now, so the first thing they did was to actualize this experiment. What they did was to find a capsule, because when they did the isolation, they found this bacterium was in a capsule. In a capsule, it's like a round substance where you have different stuffs in it. Let me put this pointer on. So here we can see a capsule. And you remember we talked about the S strain and that of the R strain, right? So the S strain is the smooth strain, right? And then the R strain is the rough strain. One of them is lethal and the other one is not. So let's try to see which one is lethal and which one is not. By this point, people were still investigating to know what was the transforming principle. When you understand what is the transforming principle, then you carry on another experiment to know if that transforming principle is a protein or is a DNA. So at this point, we want to know what is causing this infection. If we get to know that, then we will proceed on to investigating or finding out whether that particular thing that is causing the transformation is either DNA or a protein. So our first investigation is to know what is a transforming principle. Now, in doing that, we have the S string as well as the rough string. So here we know that the S string, like I said previously, we discussed this, the S string, it just seems so redundant to explain it again. The S string is the smooth string and it's the lethal string, right? So here they extracted the S string, injected the mice and it died. And in the next experiment, they took the R string, which is the rough string. R is there for rough. And that is because if you look at the bacterium, if you look at the string that was isolated from the bacterium, it has some rough patches on its outer portion. So they injected the mice and it lives. So what they did was they collected the smooth string here, this blue one or purple, they heat killed it. So that means, in microbiology labs, usually you find people heat killing things, right? They call it sterilization or, to kill heat by heat killing. So they heat kill it. After heat killing it, they injected the mice with it. The mice did not die. So that indicates there was something in this particular rough string that was alive, that had life, that is carrying on, some form of transformation. And the life that this thing had to carry on transformation was actually the genetic material. But what it is, we don't know yet. So that's what we're trying to investigate. Now we have this smooth string, we heat kill it, we inject the mice, it lived. So which means this whatever that is here that carry on this transformation can be killed by heat. So that means it is vulnerable to heat, right? Now, and in the next experiment, what they did was they took the heat killed string and then they took the rough string. Remember the rough string is in this orange or yellow, whatever it is. We have this orange and then we took the heat killed one. Remember, heat killed doesn't kill the mice. Rough string doesn't, heat killed smooth string doesn't kill the mice. Rough string doesn't kill the mice. But then they took the smooth string, they heat kill it, right? They heat it, which means dead, doesn't kill the mice on its own, and they took the rough string, which on its own doesn't kill the mice. They mixed the two of them. So if you look in here, you see it's a mixture of the heat killed them. When they injected the mice, it died. So what happened here now, there was a transforming principle. Although the idea is that although the rough string doesn't kill the mice on its own, but whenever it is mixed with the smooth string, it does transform itself from the rough to the smooth string. You see, so it transforms. So that's a transforming principle. And we know in molecular biology, transformation takes place. You have the ability of converting DNA to RNA and RNA to proteins, and so on and so forth. We have these things in molecular biology. Living organisms have the ability to convert themselves into different things whenever they want to carry on some form of please mute yourself, stop the video, mute yourself. So they have the ability to do that. Now, we also have the live S string was retrieved. So during this mice, they carry on some form of extraction. After they extracted, what they retrieved from this form was the live S string, which means the smooth string in the presence of this heat-killed, the rough string, in the presence of a heat-killed smooth string, it transformed. Although this one was heat-killed, it died, but that means not everything within it was destroyed. If they are combined, they will transform. So that's what happened. That was one of the experiments that they realized that there is something going on. There is actually something that is carrying on this biological process. That was what Mr. Griffith covered. Please mute yourself. So that was what Mr. Griffith discovered in this whole experiment. He realized that, yes, there's a transforming principle, but what it is, we don't know. We still don't know what is transforming principle, but we know there is something that is causing that. Okay, so now after realize this, and then, it was done sequentially. If you look at the previous one was in 1931, right? And then the next experiment was in 1952. That means 20 years or 21 years after. Another experiment was carried on by Alfred Hershey and Martha Chase, or also they call it the Hershey-Chase experiment, right? What these guys realized, of course, this experiment was based on the previous experiment that was carried on. There's a transforming principle. So Alfred Hershey and Martha Chase also used what we call the T2 bacteriophage, which is a bacteria that is used to, transport other things. Bacteriophage is a kind of virus that eats a bacteria, right? It injects and destroys bacteria. So it is a revolutionary bacteria currently that is used in a lot of experiments. It's very, very important, right? And now scientists have the ability to take this bacteriophage, this guy with a big head, and remove this genetic material from in it and put their own genetic material of interest. So this is its DNA that is inside. All behind it is just a capsule or capsid. It has a tail, a fiber, and this is a bacteria, which is called E. coli, right? The cytoplasm of the E. coli or Escherichia coli. So what they did here is to take a DNA and then place it in this bacteriophage and allow this bacteriophage to infect this bacteria. And then in the next part of the experiment to realize what is going on. So let's see what happened. Remember, in the first part of the experiment, this is the mechanism that takes place when it comes to carrying on in this experiment, right? They allow naturally or normally, bacteriophages are always infecting bacteria. This is a normal process. It happens in nature. It is not induced by human, or humans have taken advantage of this and now using it for experimental purposes. So this is an old technology. Now that it was being discovered, by then these people decided to do that when they realized bacteria cells that. So Hershey Chase showed that DNA, not proteins, enters bacterial cells and directs the phage reproduction. Now, I want us to go back again and see this guy. The whole part of this, besides this one line that is in here, this one line is the DNA. Outside of this line, it is the protein. Protein is different from DNA, okay? In a sense, because their functions are different. Of course, proteins come from DNA, but protein is different from DNA. It is when DNA gets translated, it becomes protein. But once it is not translated, it is not protein. Okay, so now proteins do not carry proteins, they are not the ones that carry on the transforming process. It is the DNA, because the DNA contains the exact information. All right. And eventually you have my whole body right here, and you want to know what kind of knowledge do I have? And to know what kind of knowledge do I have, you need my brain. My brain is where all of my knowledge is stored, right? You can cut off my hand and then you say, I want to see how Mr. Jallo is intelligent by cutting off his hand and now go and experiment on his finger. No, he can't. You know my intelligence based on my brain, right? So that's the same thing with these bacteriophages. Okay, so what they did, Hatcher's first experiment of the virus DNA label, right? Here we have the bacteriophage, right? It is right here, it's labeled. We have the DNA that is labeled with 32 phosphorus, right? And that is in yellow. So we have the DNA right here, and we have the bacterium. So in the first experiment, when bacteria and viruses are cultured together, right here, they are in this culture, radioactive viral DNA enters the bacterium. So they took the bacteria, they place it in this particular beaker, they culture it for some time, and they took the bacteria, place it in there and allow the phage to infect the bacteria. When the phage infected the bacteria, and then look at it, the colors are different. All right. We have 32P and 35S. 32 phosphorus, which is in yellow, and 35S, which is sulfur, which is in blue. So we colored, the scientists colored this particular bacteriophage in two. They colored the protein capsid with blue, and then the DNA in yellow. They allow it to infect. When it infected, they took this particular stuff right here, the bacteria, they place it in a blender. When they place the bacteria in a blender, they allow it to blend. Blenders are actually things that, blenders are actually things that, mix things together and then allows them to mix up homogeneously. So when it mix they carry on the centrifugation process, and what they realize that the virus in the liquid are not radioactive, right? But here they realize the bacteria in the sediments are radioactive. Now, when they look at this bacteria right here, they saw this particular DNA inside. Remember, the DNA was yellow, and the DNA was the one that carried transforming principle. So what they realized is that all of the protein part of the DNA, all the protein part of the bacteria left outside, and whatever that enters inside was the one that carried transformation. So it was the DNA. These are all experiments that have been carried out over and over to try to understand if DNA was actually a transforming principle, right? So the reason why this 32S or 32P is a dye, is a label that is used. So they'll take it and attach it because it has high affinity for DNA. So that is why they use the 32P phosphorus. And then they have the 35S, which is sulfur, that has a high affinity for protein. So they use that to attach it to the protein. And then when they allow the bacteria and that of these bacteriophage to interact, they took only the bacteria because there is a certain amount of time that they need to be together. The bacteriophage will eventually infect the bacterium. They take only the bacterium, put it in a blender and, grind it, put it in the centrifuge, separate it, and then try to monitor the bacteria to see what is inside. Is there the protein or only the DNA? And what they found was only the DNA was in the bacteria and not the protein. So the protein left out. We can't see it with our eyes, right? We can't see it with our eyes. That's why we need these dyes to help us visualize it. Okay. If you remember in our introduction, we spoke about that, the importance of having dyes to monitor cells, right? Okay. So now they knew DNA. That was the beginning of understanding that DNA was the transforming principle. So please, Microsoft, now that we know DNA is the transforming principle, let's get to know, now that we know DNA is the transforming principle, and let's try to understand what are the building blocks of DNA, right? So nucleic acids are the ones that make up DNA. All right, nucleic acids. And DNA is a polynucleotide, which means it have many, many different nucleotides joined together to make it up. When I say poly means many, nucleotides means, nucleic acids joined together. That will make them nucleotides. Nucleic acids are one, and nucleotides are many of nucleic acids that are joined together, right? Now, each nucleotide is composed of 3 parts. Each nucleotide is composed of 3 parts. Remember, we have nucleic acids. Nucleic acids are the building block. Nucleic acids, we have the 2 versions, right? We have the 2 types of nucleic acids, right? Remember 105, 106 lectures. I'll leave some of them for you to think about. We have the DNA. The DNA is a polynucleotide, which means polymers of nucleotide. Poly means many, right? Mono means single. Poly means many. Polymers, when I say polymer, which means it's joined together, all right? Polymers, when I say polymers, which means different types joined together, like you take a brick, small block, then you join it together, or a chain, they are joined, all of them are joined together. That's what makes it the polymers of nucleotides. Plenty of nucleotides joined together. Each nucleotide is composed of 3 parts, right? Now, these 3 parts are, one, they have a carbon 5 or C5. That means 1, 2, 3, 4, 5 different carbons that are called a deoxyribose. We are discussing DNA. Someone will say, "Oh, what about the ribose?" Of course. So yeah, you may be right. So the C5 is a sugar that is called a deoxyribose that is only unique for DNA. If it is for RNA, we know it is a ribose sugar. Or since we are discussing DNA, we'll call it a deoxyribose. But the one unique feature for both DNA and RNA is that they both have this, the carbons are 5. One of them is not 6, the other is 5. No, they both have 5, which is a ribose. Ribose means 5 carbon sugar. Now, because it's a DNA, so we say it's a deoxyribose, okay, which means one portion of that particular ribose sugar, a hydroxyl group has been removed. We only have the hydrogen part. That is one. And the other aspect, the other component of the nucleotide is a phosphate group, which means it has a phosphate group, right? We have a nitrogen containing base as well. Now that we have named all of them, let's try to point them out. If you see, you can see an image on this side. So we have the C5 sugar called the deoxyribose. So if you look here, you notice that this is the C5 sugar. We have 1, 2, 3, 4, and 5. So you see 5 carbon sugar deoxyribose. And then we have a phosphate group. The phosphate group is this P right here with this plenty O's and hydroxyl group attached to it. That's a phosphate group. And then we have a nitrogen containing base, right? Please mute, please mute and go away. I don't want to see you. So, and then we have a nitrogen containing base. The nitrogen containing base is the one right here. Here is the nitrogen, yeah, the N. And then we have this whole base, a nitrogen containing base. And this nitrogen containing base can have different types. We have the purines, and then we have the pyrimidines. Remember, right? So I'm going to leave that with you again to refresh your knowledge. Now we have our DNA right here. This is a polymer now, polymer of nucleotides. So this is a nucleotide on its own. This is a guanine. So now the nitrogen containing base part of the DNA is what differentiates the different types of nucleotides. Here, you may have a guanine, you may have a uracil, right? You may have a thymine, no, not just DNA. Let's just say, genetic material, let's say both DNA and RNA, you know, there are 5. If you combine DNA and RNA, but there are 4 only for DNA, 4 only for RNA. And then you know what makes them different, because we already talked about that again. What differentiates DNA and RNA is just one of the nucleotide, right? One is available in the other and it's been replaced by another one in the other as well. So this is a guanine, which is a G, and this guanine is a nitrogen containing base. And then we have the ribose sugar, 1, 2, 3, 4, 5. This is the S, which represents the sugar. And then we have a phosphate group right here. So 1, 2, 3. And then we also have another one right here, another nucleotide, which is a thymine. We have another one, which is an adenine, and this is a cytosine. So what you did not see here is uracil. And why there is no uracil is because this is a DNA. If it is an RNA, we know that we'll replace thymine with uracil, right? We'll replace thymine with uracil because we will be discussing RNA. But since we are discussing DNA, we have thymine and not uracil, or uracil, like you call it. And DNA has 2 parts. They have the 5' end and the 3' end. And this is called a polymer of nucleotides, or a polynucleotide. Like I told you, they call them poly because you have all of these guys joined together. You can see guanine and thymine are joined together by this bond. It's called a phosphate bond, right? Or DNA, often you hear that they say DNA is joined by a phosphate backbone. So these are the phosphates here. Phosphate, 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 phosphate. So that make up their backbone. That's a vertebral column right there. And then this DNA is one strand. This is a single strand. You have another strand right here connecting another part of the DNA as well. I hope this is clear. If you have questions at any time, you can stop me. You can just raise your hand. I'll acknowledge you and then you ask a question. So let's talk about the nucleic acids, the different types of nucleic acids. There are 4 possible nitrogen bases in molecular biology, or in making up DNA. There are 4 nitrogen bases, right? We have the adenine, okay? We have the guanine. This is an adenine, this is a guanine, and they both make up what we call the purines. And one smart way to remember them is that you should realize that the purines have double rings. So we have the small baby ring and the larger one, small baby ring and the larger one. And the difference between adenine and guanine is adenine has an amino group, whereas guanine has a carboxyl group at this point. Okay, that's also something you try to remember. And then for the pyrimidines, it has thymine and cytosine. They both look so much alike. But again, the difference here is that one thymine has a carboxyl group, and then cytosine has an amino group. So that's the difference. So these are the 4 building blocks of DNA, not RNA. These are DNA, okay? Now, let's go to RNA. Nucleic acids are made of RNA or ribonucleic acids. Ribonucleic acid contains a 5-carbon sugar called ribose, and the bases are adenine, cytosine, guanine, and uracil. I told you before, uracil will replace thymine in RNA. And what is the difference between the DNA and the RNA is that a major difference is not just the replacement of the uracil, it's also that on the sugar ribose, we have at this carbon. For DNA, we have only H, no O. All right, but for RNA, we have the OH. That's why DNA is called a deoxy. Deoxy means remove this oxygen, you only have hydrogen. But for RNA, we say ribonucleic, no deoxy, which means we are not removing anything. We are not removing any O from there, so it remains. Besides these two, besides the availability of uracil and that of the presence of this OH group right here, RNA and DNA, it is single-stranded. Of course, you remember I told you that double-stranded RNA, they are single-stranded DNA, but we are not going that level right now. The time will come, we'll discuss that. But right now, one of the fundamental things we might want to remember is that, oh, RNA is single-stranded, DNA is double-stranded. But when we go advanced, when we go to the bigger levels, you notice that there are some that is single-stranded, and then there are some RNA that are double-stranded, okay? So these are the major differences. But besides that, everything else remains the same. It has a nitrogenous base, it has a sugar ribose, and then it has a phosphate group, the 3 basic elements of all nucleic acids. Okay, so let's go to the next one. DNA is a functional genetic material as one varies between species and individuals. DNA in one organism is different from the DNA in another organism in their functions. It is not in their makeup. They all have the same makeup, but functionally they are different. Okay? And there are also, DNA is a functional genetic material as it can store information. DNA can store information. DNA remains constant within a species. This is not the case all. DNA sometimes in some species can carry on evolution, and when evolution happens, some part of your DNA changes. So this is not a Bible verse, so it's not a Quranic verse, it's not a Holy verse, okay? It's what been observed elementarily. We can say DNA remain constant within a species over time, but due to evolution, sometimes your DNA can change, it can shift, okay? But when I change completely, it's just a little bit of tweaking, just to edit a little bit. DNA can replicate, and DNA can also undergo mutations. Okay. Now, let's look at the structure of DNA. Now, DNA is composed by double strand of nucleotides, where C always pairs with G and T pairs with A. So, we have 10 more minutes. So, the C, which is cytosine, always pairs with G, guanine, and T pairs with A, adenine. And then we also have these two, yeah, that's what happened. And whenever guanine is pairing with cytosine, we have 3 bonds. Please remember that. The pairing between guanine and cytosine have 3 hydroxyl, hydrogen bonds. And then the adenine and that of thymine have 2 bonds. That is very, very essential to be remembered. Now, the discovery of the structure of DNA. There were a lot of contributions that were made, and many of you have heard about, Madame Curie, all right, the female scientist that was treated well by James Watson and Francis Crick. But now she's been given the credits she deserves, all right? Hopefully, scientific community can forgive James Watson and Francis Crick. So the structure of DNA, these are the individuals that contributed to understanding the structure of DNA. And some may want to say, why is it important to understand the structure of DNA? Well, if we don't understand the structure of DNA, we will not be able to understand the function of this DNA as well. So it's very, very essential to understand the structure of DNA. That's why, in order to understand how DNA functions, or how to know whether there's a mutation or not, or how to know if there's an evolution, you have to know something to know if it's changed or not. Look at your community. If you don't know your community, you will not know if there is a change that occurred in that community, right? If you don't know yourself, you'll not know if you are evolving, if you are changing, if you are growing, right? So that's the same way. we needed to know the structure of DNA to be able to study DNA very well, to realize how DNA is different from one animal to another, from one person to another, how animal DNA is different from that of plant DNA. We needed the structure. So these individuals contributed to that. And this lady right here is called Rosalind. I mean, not Marie Curie, sorry. Curie is, no, this is Rosalind Franklin. Rosalind Franklin. Rosalind Franklin is the one, well, she was a X-ray, X-ray diffractionist. So she'd use X-ray beam, right, to diffract crystal and to know the structure of DNA. And right here was one of the pictures that she got. So every time she takes some, when she extracted the DNA, you remember we already discovered that yes, DNA is the genetic material. Now we need to know what is the structure of DNA. After realizing that DNA was a genetic material, after a series of experiments, the next one was to know, okay, what is the structure of DNA? Because that's how it evolved. Now, knowing the structure of DNA will also give us more advantage in studying it and understanding what are some diseases that are associated with it. So this picture is called photo 52, right? Photo 52 was the 52nd image that Madame Franklin had taken from her experiment, and she placed it somewhere. And James Watson and Francis Crick were the ones that visited her lab, one of them, and took this image from her desk without her consent and took it. They used it to solve the structure of DNA, to know that DNA is, anti-parallel, and then it's here and it's pairing and so on and so forth. So here you have James Watson and Francis Crick, the two of them. In Watson and Crick double helix model, weak hydrogen bonds between the bases hold the two chains to one another. They knew some of the bases, but they were confused on what bring the two helixes together, right? One end of the DNA and the other one. So with that picture, they were able to decipher that and understand that yes, DNA was carried by this way. Okay, DNA is suitable for replication. One other area of DNA. So we'll just try to go through this. We'll go through them in detail, how DNA replication occurs and so on. DNA can undergo a process called replication. That is, DNA duplication is a semi-conservative as each new filament contains an old strand and a new strand. So DNA can go through the process called replication. It divides and gives rise to a new DNA. And DNA is suitable for replication by three processes, right? Unrolling and winding, complementary base pairing, and joining. DNA polymerase is the enzyme that assists in step 2 and 3. DNA, this is the same thing about the DNA replication process. Hello? I have a question. So we will go through these things in detail, the replication of DNA, transcription, translation, all of them will have more details to go through them and as time goes. So DNA, we also have DNA polymerase, which is an enzyme that is responsible to help duplicate DNA, right? We have DNA replication right here. The second strand of DNA is duplicated in opposite direction of the replication fork. Replication is continuous for the leading strand, but discontinuous for the lagging strand. We'll also talk about that in more detail. Genes and protein synthesis. This is the part that we call translation, all right? But before translation, you know that we have transcription, right? To produce an mRNA, and that mRNA will now be translated into any kind of protein that we need, okay? Protein synthesis this is what we call gene expression. We have the transcription and translation process of DNA. DNA is transcribed into mRNA. In translation, the transcript of mRNA directs the amino acids sequence. That is, we discussed this also in our 105. Okay, this is the genetic code that we also discussed about. The translation process highly depend on this. It's just like you, when you are given a letter to read and you don't know some of the definition of the words that are there, you take your dictionary to try to understand what they mean, right? So also the translation process also uses this particular dictionary to be able to carry on to know the nucleotides, how many nucleotides or what type of nucleotides correspond to which amino acid. The transcription, during transcription, complementary base pairing occurs. RNA polymerase join bases and the coded information passes from gene to mRNA. We have the transcription process right here. We have the introns, that is, post-transcription modification. We have the translation. This is the tRNA is the one that is responsible to bring the amino acid to the mRNA and then convert that nucleotides to an amino acid. We have the ribosome. We have the small and large unit. We have the prokaryotic and the eukaryotic ribosomes, right? They are different. We have one which is an 80 and the other one is a 71. So we'll discuss them also in detail. We have polypeptides. Polypeptides are things that have been synthesized. Like I told you before, until after translation, until the transcript, I mean, these amino acids, until they can take their three-dimensional structure, they are not proteins. They are called polypeptides. All right. After they have been translated, they're released from there, and then they take their three-dimensional structure, then they become. Mute yourself, please. Okay, so they take their three-dimensional structure. Okay, now polypeptide synthesis step one, the initiation, which begins the process. And we also have polypeptide synthesis step two, gene expression, mutation, right? Point mutation. We discussed all of these. Frame shift mutation. We didn't talk about transposons, right? By Barbara McIntosh. Barbara McIntosh was someone who was studying corn, and she discovered this particular thing called transposons or jumping genes. It's a sequence of DNA that can change position within a chromosome. So they have the ability to change their own position without any permission. And in most instances, this calls for evolution, or sometimes it causes for mutation, right? It's both harmful and helpful. So we will just try to give you to read the rest of the notes, and then our next lecture time will begin from there. So stop the sharing and stop the recording.